Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the fifth webinar in the Neurophysiology 2023 series. Today's webinar is titled Evaluation of Novel Therapies Using Spontaneous Seizure Models, featuring Dr. Cameron Metcalf, Research Assistant Professor of Pharmacology and Toxicology at the University of Utah, and Katrina Irie, CAHA Sales Specialist with 80 Instruments. Today, Dr. Metcalf will cover the technical requirements and benefits of spontaneous seizure models, addressing the challenges and optimization of 24/7 video EEG data collection for epilepsy therapy development. Additionally, Katrina will provide a demonstration of the LabChart software from 80 Instruments used to capture EEG data collection along with many other signals. Before we get started, we'd just like to acknowledge our partners for the series, the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd like to give one final special shout out to today's session sponsor, 80 Instruments. 80 Instruments creates industry-leading software and hardware tools for life science researchers and educators. For over 30 years, 80 Instruments' mission has been to support scientific discovery, and they're proud to be in strategic partnership with the American Physiological Society to support innovation in the life sciences. 80 Instruments offers a range of solutions for accurate physiological measurements, and with the ability to integrate multiple data sources, systems can evolve as your experiments do, ensuring quality results wherever your research takes you.
Thank you very much, Sydney. I'd like to thank the organizers for this meeting for inviting me, 80 Instruments, the American Physiologic Society, and the other sponsors and administrators of this meeting. It's my pleasure to be with you today and to tell you a little bit about the program that I work with here at the University of Utah and the College of Pharmacy. The title for the talk today is the evaluation of novel therapies using spontaneous seizure models. So I will be speaking about animal models of epilepsy and of seizures today, and I will have some videos for that as a disclaimer going forward. I'd also like to mention there are a few acronyms that I'll be using. My position here is with the ADD, Anticonvulsant Drug Development Program, which is the name that we're known by at the University of Utah, of course, and throughout the country, through our collaborators and through the years. This has historically been the way that we've referred to ourselves as the ADD program. I'm the associate director of this program. Our principal investigator and director is Karen Wilcox. I'm also a research assistant professor in pharmacology and toxicology here at the University of Utah. I'll begin with just some disclosures, and I'll also introduce a few of the other acronyms today. I'm going to make reference to the NIH/NINDS Epilepsy Therapy Screening Program, or ETSP. And while I'm not representing that organization today, I will make reference to it in my talk. Okay, so our goals today are to first understand the animal models that are used for screening of novel anti-seizure drugs. We are primarily a screening organization here within our lab at the University of Utah, and that will be primarily the direction that I address my comments with today. I'm also going to specifically focus on some of our capabilities of using video electroencephalogram or video EEG for the monitoring and evaluation of spontaneous seizures. So briefly, for those that may not be familiar with epilepsy, I'll introduce this as a family of brain disorders that cause seizures. Epilepsy as a diagnosis can come from a variety of different potential causes, and it has a number of different seizure types and overall etiologies. It can be caused by genetic factors. It could be monogenetic or polygenic. Environmental insults, such as infection, inflammation, traumatic brain injury, or commonly for unknown reasons or subclinical combinations of what I've mentioned already. This affects approximately 1% of the United States population. Many of us in the audience today will know or have known someone with epilepsy, or have it ourselves, or have contributed to care for someone with epilepsy or with a seizure disorder. Epilepsy is treated by one or more anti-seizure medications, which I've abbreviated here as ASMs. It can also be treated with implantable devices, such as a vagal nerve stimulator or other stimulating devices, or with a surgical intervention, such as resection of tissue that is known to cause or promote seizures. My comments today are going to be primarily centered around the use of anti-seizure medications. Importantly, as it relates to that, we know that approximately 1/3 of all individuals are refractory, which is to say, do not experience full seizure control on one or more anti-seizure drugs. And so it may be the case that a patient starts, once diagnosed with epilepsy, on one anti-seizure medication, and it may be that that is not fully effective in reducing seizures or has notable side effects. And so additional medications may be attempted or combined. And we're seeing that as long as we've been tracking this, and so that it continues for at least the last 2 decades, about 1/3 of our patient population experiences this pharmacoresistance. I mention it here because it's a major area of emphasis for our program. We are primarily focused on seeking, helping to develop, identifying new therapies for the treatment of pharmacoresistant epilepsy. So I'll mention a little bit of history with respect to epilepsy drug development. Here on the graph on the right, we have a timeline, and this comes from a recent review article. And this timeline shows the approval of a number of different anti-seizure drugs. So starting about a century ago, we had just a few compounds available for the treatment of epilepsy. And you can see in the early 1900s, a pretty steady increase in the number of drugs that were used for the treatment of seizures. And this is the first and second generation of anti-seizure medications. Around the 1970s, there was a bit of a stagnation in the development of new anti-seizure drugs. And as part of a public-private partnership, the Anticonvulsant Screening Program, or ASP, was formed at the National Institutes of Health, NINDS. And that program continues today, and the way that it was administered is with the contract, wherein the contract site, which has been held at the University of Utah since 1975, has continued. And so drug developers, for example, can submit their compounds to the NIH after going through an approval and vetting process to see if their compounds, drugs of interest, are appropriate for the current goals of the program. When submitted, they are tested in a blinded manner at the contract site, again here at the University of Utah. And that program, as I mentioned, continues today. It is now known as the ETSP, Epilepsy Therapy Screening Program. And not shown here, but we have contributed in major and minor ways to a number of the drugs that have been approved since the inception of the program. So this third generation of anti-seizure drugs, most recently cenobamate, which is at the top of that list. During the early phases of drug development for epilepsy, I refer to them here as the early workhorses, were maximal electroshock, which is an electrically induced seizure, administered generally to animals that are drug and seizure naive. And a drug is administered prior to the administration of that maximal electroshock. And then observations are made as to whether the outcome, which is to say the seizure observation, is different in the presence or absence of drug. Pentyline tetrazole is a chemically induced seizure model as well, and that has been heavily used. We've also begun to use kindling models several decades ago, and now more commonly, and again as the major subject of this discussion today, will be the use of spontaneous seizure models. Because of their etiologic relevance, we feel these are becoming increasingly more important, but require different methods and infrastructure to be able to properly run these studies for drug screening. So I have an overview here of a number of different animal models. This is by no means comprehensive, and this does heavily weight rodent seizure models, although of interest there are other models that are captured here, and some that are not shown here, such as zebrafish models, which have become increasingly important and widely used in our field. In general, on the left-hand side of this diagram, you'll see reference to genetic animal models. With either spontaneous or induced seizures. And today I'll show you an example of animals with a mutation in a sodium channel that have recurrent seizures and how it is that we approach monitoring of those animals. On the right are the induction of seizures in order to model epilepsy. And as I've already mentioned, that can be done acutely, which is to say in animals that have not yet experienced seizures, and in drug-naive animals as well, either with maximal electroshock, 6 Hertz induced seizures. Generally, these seizures occur when an electrode is placed on the corneas or on the ears, and electrical properties sufficient to generate that type of seizure are applied, currents rather applied to produce those types of behavioral seizures. Similarly, chemical seizure induction can be used. So the example here on the right is PTZ or fluorothil, PTZ being pentyline tetrazole that I mentioned before. These can also be used or adapted to produce a chronic seizure state. And so there are 6 Hertz kindled models or other corneal kindled models, for example, amygdala kindling models. And what this is the application of a very small stimulus, which is to say subthreshold, subclinical stimulation. So a very small dose of pentyline tetrazole, for example, or a very small amount of current that's applied. Initially, that doesn't produce a seizure, but over time it does after daily and repeated stimulation. And so therefore, those animals are considered kindled. Importantly, though, as I mentioned, we are using models of spontaneous seizures. And in the case of what I'll talk about today, post-status epilepticus models, and I'll explain a little bit more about that, where the animals have spontaneous seizures, and we feel that this more closely mimics what patients with epilepsy are experiencing, and it aids in our drug screening efforts. So how is it that we approach the onboarding rather of models in our program? So first we like to identify a need. So the best example of that is as we are interested in identifying novel drugs for pharmacoresistant epilepsy, the need there is for models that appropriately reproduce that drug resistance. And so we have been focused on animal models where when we apply a number of different anti-seizure drugs that are currently available, FDA approved, many of those drugs do not work at blocking seizures in some of the models that I'll talk about. And so there's a need there, and that has informed some of our model development optimization effort. So we go through the process of developing our methods, and that can include optimizing a standard operating procedure or SOP. We are also beginning more and more to include common data elements into our case report forms to help in our rigor and reproducibility. And so that is instituted at this method development and optimization stage. And then ideally, we can do some kind of pharmacologic validation, which is where ideally several drugs comprising mechanistic classes within the epilepsy space are evaluated in the model. And in the example that I gave, in general, what we would like to see for a new model of pharmacoresistance is that most drugs that are currently approved don't necessarily work. And so we see that in the rat 6 Hertz model, which is not a spontaneous seizure model, but it's an induced seizure model in drug-naive and seizure-naive animals. And we developed this model because there needed to be a rat comparator for the mouse 6 Hertz seizure model. We saw this as a need, and again went through this process where we developed the model, optimized it, and tested a number of different compounds, and that reference is shown here. Similarly, using an ex vivo model, looking at brain slices with spontaneous seizure-like activity, we use this approach in a similar manner and develop this as a means to screen novel anti-seizure drugs. And finally, here for this slide, I'll also mention one of our kindling models wherein lamotrigine, the anti-seizure medication, is administered during the kindling process. And during that kindling process, when lamotrigine is administered, when the animals become fully kindled, they are also resistant to a challenge dose of lamotrigine and, in fact, resistant to a number of other anti-seizure drugs. And again, the goal here for our program is being focused around pharmacoresistance, and so we found these models to be very useful. Now I'm going to turn my attention to speaking about spontaneous seizures, how we evaluate those in our rodent models, and what some of the infrastructure, techniques, methodology is to do that. So I will come back to this slide at a couple of different points, but I just wanted to mention some of the key elements here for being able to evaluate spontaneous seizures. Historically, and for many years, and I can say even when I was in graduate school, EEG was used, but we also just use video and to look at behavioral seizures. Now we have the capability of combining those two things. So what do you need in order to establish a program or a recording unit to have this type of capability? One is an experienced a surgeon, somebody that knows how to implant devices, knows how to implant electrodes, properly secure them in place. As you might imagine, since we're working with rodents, we don't have very much control over how much they interact with their implant. And so a skilled surgeon and a skilled surgical approach is required. For tethered EEG, which is what I'll be talking first, the animals are connected via a wire, and then that wire is then, by extension, connected to our recording equipment. And so that requires a commutator on the cage that allows the animal to be more freely moving. And again, in order to capture the video EEG, those impulses, need to be synchronized. And when synchronized with video and EEG, and I'll give an example of that in just a minute, we have the ability to look at electrographic and behavioral seizures. Amplification of the signal is required. I'll reference this a little bit later, but as we are reviewing seizures, automation, whenever possible, can be really beneficial. So on the lower left of the diagram, I've mentioned here expert reviewers. It's very important to have individuals that are looking at electrographic and behavioral seizures that have experience in recognizing some of those signals. And I'll give an example of that in just a minute. But as part of that, whenever possible, if automation can be used to identify seizures ahead of time so that they can be evaluated rather than going through the video and electrographic signals temporally, then that really helps with the overall efficiency of this type of analysis. So I'll turn briefly and mention one of our genetic models, and this is an animal model that has spontaneous seizures, and it's a model of Dravet syndrome. Dravet syndrome is a rare type of epilepsy that is generally due to a mutation in the SCN1A sodium channel. Here's an example of what that, a diagram rather, of that channel. And in our animal model, we use a point mutation model where the animals have seizures, pharmacoresistance, some behavioral comorbidities that we've been able to evaluate, and a high mortality rate, which, to the second bullet point here, mimics what is seen in patient populations with Dravet syndrome. So in addition to other models of Dravet syndrome, mouse models have been used to study the pathophysiology of Dravet syndrome, morbidity, and anti-seizure medication pharmacology, rather, for this model. So here's an example of one of our tethered units. Before I start the video, what you'll see here is an animal that is connected from its head cap, which is dental cement, holding in place surface electrodes. And connected by a wire that connects to a commutator. What you can't see on this is where the wires go afterward, which is again getting toward the amplifier and other hardware that helps us to be able to record multiple animals at once. You'll see at the top of this diagram that we have an electrographic seizure. And then the video that I'm about to show will also be timed so that we can see the animal's behavior in tandem with that signal. So as the recording begins, the red line is moving at the top across the EEG trace. And then the animal below is normally behaving. The seizure will begin in just a few moments, where we'll see forelimb clonus here beginning, some rearing up. The animal will fall over. You can see the sort of rapid movement here that's beginning to coincide with high frequency, high amplitude behavior or activity on the EEG trace. This animal's seizure behavior will continue and progress to some extent. We'll see in a moment that the animal will kind of jump around and run a little bit, which is a more a later version rather of this seizure. Now here, as we're reviewing this EEG, and if we come to a signal that looks like a seizure, we can now go to the video. And in our models, that helps us to verify what type of seizure has occurred, the extent to which it occurred, its duration, even if that's desired, and overall severity. You'll note here at the top what I'm showing is an example of an EEG trace. But for those that may have experience with EEG, it should come as no surprise to you that sometimes what we're faced with are signals that have more noise in them. And that's particularly the case for tethered EEG. And it's something that we've had to contend a fair bit with. And this gets back to my point about needing experienced reviewers that can look at both the EEG trace and some of the subtle behaviors that show up for some seizure types in these animals to recognize whether something was a seizure or not. And as you might also imagine, given the noise that may occur in a signal, that makes automation a little bit more difficult. So for any electrographic type of signal that is used, signal-to-noise ratio is very important and very beneficial when it can be minimized. And so I'll move now to our status epilepticus models. Again, spontaneous seizure models. And for those of you that are less familiar, I'll tell you that commonly canic acid or pilocarpine, there are other agents that are administered to rodents, produce a period of acute seizures that are repetitive and can continue for minutes to hours. And that period is referred to as status epilepticus. Following that time in rodents, and it's been well established, there is a latent period where there are few, if any, seizures that occur, and then eventually the onset of spontaneous recurrent seizures. I mentioned here also that the timing can be variable. So for our mouse intraamygdala kainate model, for example, that time can be on the order of days to weeks, whereas in our rat post-kainate model, the period for spontaneous seizure development can be weeks to months. So there is some variation there, and again, it is model dependent. Again, I'll turn to this slide and tell you a little bit more about what our approach has been in mouse tethered EEG for the intraamygdala kainate model, and I have a slide next to talk about that. We can use surface or depth electrodes. We also have an injection cannula for the administration of kainate, which may be useful. But again, the overall approach is the same with respect to tethered EEG. The electrodes and injection cannula are secured in place using dental cement and screws into the skull. The animals are placed in cages with rotating commutators. They're allowed to freely behave. Those signals are amplified and synchronized with a video capture as well. Here, I'm showing you some data from a paper that came out recently. My colleague Peter West is shown here on the lower right. He spearheaded our overall approach to the validation of this model for use in our lab. Here, canic acid is administered through a cannula, shown in panel A, into the basolateral amygdala. And then caudal and dorsal to that, in the hippocampus, we have a recording electrode placed. Currently, we are moving more towards surface electrode recordings, but I showed this diagram here to say we can also do depth electrodes. When canic acid is administered, the animals will go through a period of status epilepticus. So if you look at the right trace in panel B, the red trace, excuse me, in panel B, you'll see that electrographic activity goes up during the period following canic acid and continues for several hours afterward. Here in the blue trace, we administered midazolam, and we show that we were able to decrease some of that activity, although not shown here, it didn't really affect the long-term outcomes in terms of spontaneous seizures. On the far right, you can see an example of an EEG trace that we've broken down further in the panels below. The point here is to say that using tethered EEG, we're able to record status epilepticus in these animals. quantify as needed that period of seizure activity. And then in the weeks that followed, if I can turn your attention to the lower left panel, we're able to perform drug screening studies. So here we evaluated a cohort of animals over a baseline period of several days, and then administered either vehicle or valproic acid to the animals. And we were able to show that valproic acid and the treatment conditions that we provided decreased the average number of seizures per day. And so this is an example of a model where we can use continuous, which is to say 24/7 monitoring of animals, enroll them in studies where we try to mimic clinical trial settings and perform drug screening studies. So finally, I'm going to turn my attention to the rat intraperitoneal kainate model, which is similar in terms of the overall approach to generate seizures, as I mentioned with the kainic acid. What's notably different here is that in our rat kainic acid model, we're using telemetry. And I'll point out why we think that has been so beneficial. So what's needed in terms of infrastructure for this type of system? Again, a skilled surgeon that can place the electrodes, even if they are skull surface electrodes. Cages with telemetry pads, and I will show an example of that in just a minute. A receiver, amplifier, and these can all be purchased in a package, of course. Some can be custom made, and we've done some of that. And then seizure review and automation. And that, again, that's very important. So to come to the point, I'll show you an example of our setup here, where we have, you might be able to appreciate rats in clear plastic cages that are sitting on top of these blue telemetry pads. And these pads allow for charging of the telemeters that are placed intraperitoneally in these animals. And so these animals received kainic acid and are now in the stage of having spontaneous seizures. And so this allows us to record from multiple animals at once. And we have several of these units or similar units for both mouse or for rat. And now as we're also developing one for mouse telemetry EEG. My colleague, Kyle Thompson, has been principally involved in all of our video EEG activity over the years. Kyle is actually with me here today, so as we get to the Q&A section, he can help to answer any questions specifically about hardware or software. We use custom software for our video EEG analysis. I'm happy to address any questions about that, but none of this would be possible without Kyle's expertise. So one of the main reasons why we really like the signals that we get from our tethered array is that they have a much better signal to noise ratio relative to our tethered EEG. So using telemetry rather has really improved our signal to noise ratio. So I'll show an example of that. We're going to be focused on the red trace that's shown here in the middle of the screen, but I hope you can appreciate that we're recording from 12 animals simultaneously. We have really nice signal quality in our view, and that allows us to be able to identify seizures really efficiently. To that point, my colleague Kyle and his team developed an automated seizure detection algorithm that has dramatically affected our efficiency because with that algorithm, we're able to see where the software will identify where seizures occur, and we're able to go directly to that video rather than chronologically going through a recording. And again, that has been a really beneficial addition to this type of monitoring. And we're excited to see where it may take us in our mouse telemetry array. So here I'll show you this video. The first part of the video is not quite synchronized. So you'll see the red trace go across briefly and then it'll come back. So here we do see a little bit of behavior with the animal here in just a minute. Some early seizure behavior that's not quite shown on the EEG, but here as the animal begins to go forward and have a generalized seizure, you can see in that red EEG trace that pairs up with forelimb and hind limb clonus, rearing and falling in this animal. And again, with the telemetry setup that we have, this animal is a little more freely moving. Occasionally, we do see that a tethered system can produce damage from the animal having some restricted movement, although we do our best to try to minimize that. One additional point that I'll make about our system here is that we again try to run kind of mini clinical trials. And so here's a little bit more information about the way that we implant the device, where we place them interperitoneally and run the electrode wire subdermally, use fixation screws. And then once the animals are kind of freely behaving for a little while, we will evaluate what their seizures look like and select for animals that are having seizures. We want to do that because, again, we are wanting to run drug screening studies, and we want to enroll, like you would in a clinical study, individuals that are having a sufficient number of seizures. And the way that might look is that when we enroll animals in a study, we could administer drug orally in food or by intraperitoneal injection or other means, and evaluate the animals in a crossover study that's shown here at the bottom. So the crossover design here does assume a relatively short half-life, wherein on a Monday through a Friday, an animal will be enrolled and receive either drug or vehicle, and then the crossover will occur over the weekend, and then the study will continue. Again, if assuming a short half-life, where we know compounds may have a longer half-life, we may extend that crossover period and extend the study as needed. On the far right, I'm showing you an example of phenobarbital. The traces in red are representing the period where drug was administered, whereas in blue, vehicle was administered. And you can see this kind of mimics the crossover design figure to the left. Here we show that there were very few seizures in the presence of phenobarbital in comparison to vehicle. Something else that you might appreciate is the variety of seizures in terms of their overall frequency and regularity, even clustering. That is commonly seen in spontaneous seizure models. And for those of you observing this today, this should come as no surprise to you. It's one of the challenges that we have. And so we have had to plan and tailor our studies to try to accommodate this relatively higher variability. And to summarize kind of what we've done in this, we've screened a number of different anti-seizure drugs with this approach. And again, primarily using intraperitoneal injection, single administration once daily, or multiple administration. We do feel that we have a really good understanding of what the pharmacology in this model is. And as I mentioned before, there are a few drugs that really block seizures well in this model. And that tells us that this model has some pharmacoresistance and that we're doing better at being able to mimic what's happening in patients with epilepsy. The last thing that I'll say about the telemetry system that's really been beneficial, that we're really happy about, is the ability to reuse, recycle these telemeters. And so in keeping with what I showed you previously, we will implant the telemeters, do a seizure monitoring, run our studies for weeks or months. And then when the animal is euthanized, we can explant the telemeter. We will clean it, verify that it is still functioning properly. And then in another cohort of animals, that telemeter can be reimplanted. We have a really talented team that has worked with these telemeters for years. And we are really happy to see the use that we've had out of these. In fact, some of our telemeters have been implanted, explanted and reimplanted several times, and that has given us a lifespan for these for this material for a few years, which is again really beneficial for the type of work that we're doing here. In conclusion, I'll say here that there are a number of different epilepsy models. The reason why I mention some of the other models is to show you where we've been and where we hope to go. that chronic monitoring, chronic drug administration in animals that are having spontaneous seizures is an important path forward for us, again, from the standpoint of etiologic relevance. We want to try to help to develop better drugs for epilepsy. And automated seizure review is very important for our video EEG monitoring. And finally, as I mentioned, the use of radiotelemetry has really benefited our overall approach with respect to signal to noise, automated seizure detection, and our overall efficiency. So I just want to conclude by saying thank you to my colleagues. Again, my colleague Kyle, who's here with me today, is shown at the top of that diagram. Myself, Karen Wilcox, who's there in the middle, at the bottom of the picture, my colleague Peter West, and a number of other individuals that contribute to the work that we do in the AD program at the University of Utah. So thank you very much for your time and attention, and we look forward to your questions.
Really great presentation, Dr. Metcalf. Thank you so much. All right, and now I'm very excited to welcome Katrina to the floor. Hi, Katrina.
Hi, Sydney. Give me 1 second to share my screen here.
Thank you.
Yeah, of course. Thank you for having me today. So I'm just going to be going over our lab chart software. We have a variety of tools available in our software to help you analyze a wide range of neurophysiology data. So to get started, I'm going to go over lab chart 8. So the first thing that opens up with the software is this welcome center. On the left-hand side, you can see all of the existing or recent files that you have worked with available for you to jump into. You can also create a new one or open an existing file or an older file from your desktop. This Getting Started tab has a bunch of example files available and demo data if you want to try out an analysis technique or play around with some example data without doing anything to your own. All of these tabs up here are customizable as well, so you can organize your experiments and your projects however you'd like. Down here at the bottom, this Feature Manager button allows you to check if the software needs to be updated. So we do update LabChart 8 if new operating systems come out or if we detect any bugs. You can find updates available here. And there's also other features that you can add on, such as our Pro modules available in LabChart 8 Pro and different device enablers to use the software with other types of equipment.
Hey, Katrina, I'll just have you hide that screen sharing button at the bottom of your screen.
Sorry about that.
That's okay. Thanks. Great.
Yes. All right, moving on. The next button is this help menu. So this is like the manual for LabChart. There's a lot of really great information in here if you're ever curious how the software is making a calculation or what a calculation even does. You're able to look it up in here, and there's a lot of really good information available. The next two buttons will take you to our website. If you have any questions about the software, you can submit a support ticket, or you can check out our event center where we typically run like surgical courses or LabChart training courses to help you with your research. Okay, so in front of me here, I have a file that was recorded from a telemetry system. An EEG signal was recorded in the top channel. The second channel was an EMG signal, and then the bottom one is temperature, as you can see by the labels on the right-hand side. Some general overview things to navigate through your data. You can use your mouse to scroll along the time axis, or of course, grab this bar down here. These buttons over here allow you to expand and contract your data as well, to view them on different time scales. And then to have your data fit nicely in each window, you can click this button to auto scale it so that it fits the range really nicely. Cameron talked a lot about seizure detection, and sometimes people do it manually. So if you want to add a comment to note a seizure in your file, you can come in here into the channel and click, or you can come down to the time axis and add the comment along the entire channel by clicking this button up here. And then let's see, say you want to just type in seizure, click add our comment. And then as you go back and you continue to move through your file, you can review all of the comments that you've created using this comment drop down. So let's say you want to go to a different section of your data, you just click on that comment, it'll take you right to it. To start analyzing your data, you're able to adjust the number of channels that are displayed by going to Setup and then Channel Settings. I'll click this button down here and add a few more. You can see I have some channels that I've already created. A really common calculation in neurophysiology is to filter the data. So you can apply a filter by clicking this drop down and then clicking the filter calculation, which just went really quickly. So you click digital filter here. You choose your source channel of your data, and then we have all of these different filter types that you can choose from. I'll keep it on low pass, and you choose your cutoff frequency and click OK. I'm going to click OK again, and then you can see all of our calculations and channels have been uploaded into our chart view. Another great neurophysiology feature available in LabChart is the spectrum view. So if I come up here to Window and go to Spectrum, I'll make this full screen. You can see that I am taking an FFT of the EEG channel. And if we go to settings in our spectrum, we're allowed to adjust our FFT size, what kind of data window we want, what percentage of overlap we want to do. And then you can display different modes, whether you want to analyze the power density, amplitude, etc. All of those are available for you. And then there's a couple other settings such as color. Colors available here for you. So that's a very helpful feature. Other calculations that are available, you can do a spectral calculation in a channel as well. So if I click this drop down from the Delta channel, click on Spectrum, again, you can choose your source channel, your calculation type. And then as we know, the Delta frequency lies between 0.5 Hertz and 4 Hertz. So that's where you set your range there. And again, FFT settings are down here as well. So you can look at the percentage total power, as well as maximums and minimums and different frequencies are all available here for you to look at your data in different ways. So let's say you've completed most of your data analysis and creating your calculations, and now you want to pull more raw numbers out of the data itself. We have a feature called the data pad that I will open up, and I'm just going to tile my windows to be able to look at my raw data and my data pad at the same time. And all you need to do is come in here, and let's say you want to analyze about a 30-second section. You just click and highlight, and then you click Add to Data Pad, and all of your calculations or all of the raw data is dropped in, and you can adjust these columns to detect whatever you want. So the software is really capable of pulling out a lot of different types of information from the raw data. As Cameron mentioned, video synchronization is really important to their seizure detection. So we also have an additional feature called the video capture module that allows you to time sync your raw recorded data with a video feed. So that's available. I don't have that set up on this file with me, but that's an option that we can discuss if you're interested. Lastly, he talked a lot about seizure detection and automation. We also have some automation features available in LabChart called macros. So this is a sleep scoring macro that I'm going to run, and I'll click scoring. And what essentially this does is it's going to step by step through the file, add a comment that dictates what state this animal is in, depending on the data. And those states are also determined by another channel that I did not bring up for you guys. My apologies. So if I show these three other channels, we have wake, non-REM, and REM. Those are defined by different calculations called arithmetic calculations. This looks very complicated, but essentially it's telling the software that if this certain channel of data, which is the delta spectrum, meets a certain criteria, and the integral of the EMG channel meets a certain criteria, then you define the animal to be in a wakeful state if those both those conditions are met and are turned out to be true. So that's a really powerful example of how LabChart can help you automate your data and make your calculations a lot easier. The next thing I want to show you is our new software called LabChart Lightning. So this has a lot of great features similar to LabChart 8 with a couple of improvements that we're working on. We're also still developing this as well to continue to make it a more powerful software. So some general navigation here. If I use my mouse, I can scroll in and out of the time axis that way. And moving along the axis, I can click this overview bar to get a general preview of what the signals look like. As you can see in this top channel here, you're able to overlay signals on top of each other. Now, LabChart 8 has a maximum of 32 channels, but LabChart Lightning technically has an unlimited number of channels. This is obviously going to depend on your computer and its processing speed. You can obviously work with a lot of data in this program, but you can click and drag different signals to different channels to help you organize your data and view it in the best way possible. So if I come up to this first channel, you can see I have some calculations that are overlaid on top of my blood pressure signal. If I want to create another calculation, I just click on my signal, click Add Calculated Signal, and then we have a drop-down of calculations that we've already uploaded. These are very similar to LabChart 8, such as taking an integral, applying a filter, or doing a cyclic measurement calculation, such as like with a heart rate or whatnot. You're also able to create a custom calculation in LabChart. Oh, my apologies, clicked the wrong button. Where you click Customize up here. And this takes you to a window similar to Simulink, where you can choose different functions from our function library here and essentially drag and drop and create your custom calculation depending on what you want to be done to the data. So this is a really powerful tool to give researchers a lot of control over how their data is being calculated. Another really great feature about LabChart Lightning is the fact that you are able to record and analyze multiple files at the same time. So if I click this drop-down menu, you can see I have week 1, week 2, week 3, week 4. Those are 4 different files that were recorded at 4 different times. And I'm able to group them together in what's called a project, right? And so in order to help you organize all of this data that's available to you in this project, you can create groups and you can add subjects to those groups to help you differentiate between what signals and compare the data and the results. So you can see I had 2 subjects in my control group, and now I have 4 subjects in my knockout group. You can also add annotations similar to adding comments in LabChart Lightning. And again, if I click on this comment, it'll take me to that location in the file. You can see there's also sections of data that's highlighted. Those are called regions. So you can take a section of data to analyze it, similar to that section I created for the data pad. And then the last thing I'll show you that's very cool in LabChart Lightning is this table view. And so this takes those regions that I've created and it breaks them down between the groups and the subjects and allows you to really organize and view your data at a higher level. So you can compare, what's going on with your values in the control group versus your knockout group, and how are your subjects responding to it. There's a lot more features in both LabChart 8 and Lightning that I could go over, but I'm going to keep it at that today for the sake of time.
So with that, let's just jump right into our Q&A session. I'm going to bring Cameron back. Hello, Katrina. And myself here. Hi. Thank you both again for your presentations. Okay, first question. Have you looked into using ML methods such as deep lab cut to detect convulsion/seizures from the video?
So if I understand the question, is it, are there sort of deep learning techniques, AI related, where we can apply that? Certainly that's something that we have thought about. There are commercially available systems that are attempting to do that. And it is something that we are very interested in doing. I should say that we already have a pretty reliable technique with our rat telemetry that has really done what we've needed it to do, which is to say identify seizures in a way where we've been able to review, for example, 24 hours of a 12 subject unit within 15 to 30 minutes, identifying a number of different seizures. And we have done some validation of that to look at the overall accuracy and precision. So from that standpoint, we feel that the signals are clean enough that they've been amenable to that type of analysis. But I definitely appreciate the point that type of learning would be beneficial.
Yeah, that's very interesting. They do have a second part of the question. How long after kainate-induced kindling is the model viable? So does the spontaneous seizure state result in any long-term damage to the animal?
Yeah, so just a clarification, it's the model is not necessarily kainate-induced kindling. Those are two separate things. We are administering kainate, and then the animals will go on to have spontaneous seizures. With respect to damage to the tissue, there is damage to the hippocampus, for example, that is consistent with human temporal lobe epilepsy. That has been well established in this model. With respect to overall viability, maybe I can answer this in a couple of different ways. If viability is referring to the overall health of the animals continue to be freely moving, well behaving for a long period of time. Kainic acid can be associated with a higher mortality rate, and we do keep an eye on that. Most of that mortality occurs early on following status epilepticus or during. Related to the model in and of itself, we do see seizures that develop in a manner that's consistent with the timeline for what you would expect for the way this model's been vetted before. So there again is hippocampal damage, damage to other brain structures, and we see spontaneous seizures emerging within a few weeks.
Okay, thanks for clarifying. That's great. The next question is asking for more information on whether the tethered setup is analog or digital, i.e. head stage on animal, and how much of the difference in signal to noise between tethered versus telemetry could be due to that analog cable movement.
Yeah, so it looks like I have a colleague that's here online, Dr. Carolina Moncion, and she helped to answer this question too. But we are certainly aware of that as a possibility. What we are in the process of doing for some of our units here is to move to telemetry in mouse. And so in the future, we hope to have mouse EEG with telemetry, and our early experience with that has been it's very successful. Let me just turn to my colleague. Did you any follow up on that question about the cable analog?
I think that, yeah, I mean, obviously a head stage would work better, but that comes with a whole host of other complications, including the weight on the animal and the additional need to protect that system, which would add a ton of different complications to the system. And so really the wireless telemetry is the answer to getting good signals.
Yeah, thank you. Hopefully y'all heard that okay?
Yes, we did.
Great.
Thank you so much both for providing that info. I think, Katrina, this one can probably be answered by you. Can this telemetry technology be used in any other vertebrate? For example, fish?
That's a great question. I don't know of anyone using it in fish, and I would hesitate to recommend it. Just given the size of the implants themselves, we have seen other people use them in like hamsters and guinea pigs and rodents of similar size to mice and rats. But my worry would be how you would implant the telemeter under the scales of the fish. I don't necessarily know if that's feasible. Cameron, seems like you're nodding in agreement.
Yeah.
I would defer to your expertise on that part.
Well, and I mean, it really is about the size of the telemeter. So, in our field, most people are using zebrafish that's obviously not going to work with the size of the animal. But over time, it's reasonable that these units can continue to be miniaturized, and so there's definitely hope there. But with respect to fish, I think there are other techniques that are useful for looking at seizures.
Yeah, I think that makes a lot of sense. Thanks for your input. I think we have time to squeeze one last question in. What effect size do you expect to see for a successful experimental drug in these models? Yeah, so in our program, what we like to see is a notable reduction in seizures. And so there, so 50% or more is what is a little bit more convincing. Certainly some drugs can have a significant but not to the same extent a reduction in seizures. But we are really hoping for those kind of larger scale differences. Okay, Fantastic. And then part 2 to that question, we might as well throw it in. How many animals are required to be properly powered to see such an effect?
Yeah, so you might be able to appreciate it from some of the data that I showed, but there is a fair bit of variability when you look at spontaneous seizures. And so our group sizes are usually 12 or more. We do a crossover design that helps with that, but it requires larger groups of animals. And so 12 to 20.
Okay, fantastic. Thank you. That is all we have time for. So I just want to say thank you so much, both of you, for your time and expertise today. I really enjoyed hearing what you had to say, and I hope you had fun as well.
And thank you. Thanks to everyone for attending.
In closing, we hope that you enjoyed this Inside Scientific webinar sponsored by 80 Instruments. Another quick thank you to all of our partners for helping make the series possible, and I hope that you all enjoy the rest of your day.